Good afternoon. Welcome to Bruker's US HUPO 2021 virtual symposium, lunch symposium. I hope everyone's having a good conference. My name is Chris Adams, and I will be presenting and moderating today for you. So we have a fantastic hour lineup. Like I said, I will present as an introduction. I will then pass Dr. Shadi Ferdosi from Seer, who will present on her work in plasma proteomics and the Proteograph. And then the remaining half of the session will be from presentation from Professor Kirk Hansen and his work in plasma proteomics related to trauma as well as COVID-19. I hope everyone is enjoying the conference. I am myself. I enjoy this new feature where we actually get to interact with each other. So with no further delay, I will start my presentation to you guys. And I am sharing my screen. Okay, so I have an interesting setup here today. So what I would like to talk to you guys today about in my agenda is some of the recent advances we've made with PACER. So PACER stands for Parallel Search Engine in Real Time, and it's a GPU-enabled search engine. It's incredibly fast because of the GPUs, and that allows us to do real-time searches even in the most arduous conditions. And the example I'll present to you guys today is in the case of post-translational modifications. Additionally, we've made some nice features in data visualization. As you guys are well aware, timsTOF data is 4D, four-dimensional. Therefore, we have to have the capability to see this collisional cross-section turn. And I have some nice visuals to show you how that enables us to see much deeper into the proteome than otherwise before. I also want to describe briefly DIA passive and PRM passive as exceptional quantitative proteomics workflows, and then do a brief summary of real-time search for you guys. So here's what I'm going to do. I'm going to welcome you guys to San Jose. So we have two timsTOF pros in our applications lab, in our demo lab in San Jose. We have two great proteomics application scientists I'm showing you here, Francesco Pignatori and Michael Krawitsky. So now I'm gonna leave this screen, and I'm actually gonna take you guys into the lab itself. So here we are inside of the instrument. And what I'm showing you here is, if you're familiar with a timsTOF Pro, is our high-star acquisition queue. So I'm not going to belabor what you're seeing, but what I am going to highlight is this particular column here. So this is a PACER column, and this is where you can set up and load methods for real-time database searching of the acquisition that are being made in real time. So there's a PACER box that is sitting adjacent to the acquisition PC on the mass spectrometer. That PACER box is controlled through an interface, a PACER interface right here. Within that interface, you have, just like you would expect, ability to create search parameters. So you can load a specific database if you'd like. You can load specific PTMs that you want to search, the mass tolerances, what have you, all the common things that you would expect within a database search. After you do that, you can generate a PACER key. When you generate this PACER key, you load those search parameters that you previously created. And then there's also this PACER qualifier. This PACER qualifier determines if the acquisition should progress or not in your acquisition queue. So you can qualify that by the number of proteins you identify, the number of peptides you identify, or the number of spectra you can identify. So I'll take you back in real time to what we're doing. This is the instrument acquiring data as I present to you, and we're keying in on this welcome PACER demo, 50 samples per day. Right now we're running a 50 sample per day protocol. We've loaded 200 nanograms of a complex digest on column, and the database searching is actually happening in real time as defined by this pacer. So now I will return to my slides, and I hope to revisit our demo lab here in the very near term and show you in real time search results. So as I said, some of the most arduous searches are done using post-translational modifications. And in this case, we've been working with cell signaling technology to do this in the context of ubiquitination. As you guys are well aware, some of the limitations in database searches come when the space gets too large. And the space gets too large because of the size of a database that you're searching, enzyme specificity, variable modifications, miscleavage rates, and the number of modifications per peptide. So as I mentioned earlier, PACER is the brainchild of Robin Park from Professor John Yates' lab, and he had a company called IPA, Integrated Proteomics Application, with this IP2 GPU search engine. So instead of searching across many tens, and if you're very lucky, hundreds of CPUs, we use GPU technology to search across many thousands of GPUs simultaneously. So this gives us incredibly fast search speeds. I have a poster that I'll be presenting this afternoon. I welcome you all there, 13.01. And in the context of this talk, what I want to share with you guys is the search parameters we use for that. So it's a uniprot mouse database with very standard mass tolerances. We allow for 3 miscleavages, and then importantly 3 variable modifications: digly motif on lysine, deamidation on N and Q, and oxidation on methionine. It's a trypsin digest, and we allow for semi-specific enzyme specificity, and the number of modifications per peptide is set at 3. So all these are the settings that are defined on the PACER box for real-time search. And we want to do this in the context of translational proteomics. So in working with cell signaling technologies, we use the most difficult of all the types of samples we can, which is actually real samples. So we did this on liver, brain, and embryo from mouse, and the PTM scan ubiquitin remnant motif from cell signaling technology was used to enrich for ubiquitin modified peptides. Because the timsTOF Pro has exceptional speed and sensitivity, we like to do things in a much shorter time period than maybe a lot of the competitors would like. So all of these runs that I'm presenting to you today are performed 50 sample per day method. That's essentially a 21-minute gradient. What I'm showing to you here is 3 biological replicates of 3 different livers as enriched using the ubiquitin scan from cell signaling. So the data is generated, goes to the PACER box, is provided search results in real time, and these are the numbers that I'm presenting to you here. So what you can see is we see 2,000 proteins, not really important given that this is an enrichment. We see on average somewhere between 12 and 16,000 unique peptide observations, and importantly, there's somewhere around 3,500 to 4,000 ubiquitin peptides that are identified in real time for these samples, which is given a 21-minute gradient and 200 nanogram load. You'll appreciate that those are pretty substantial numbers. It shows that the ubiquitin enrichment rate is quite substantial, around 30%, and we're very happy with these numbers. As an example there, I want to show you from biological replicate 2 from this 1.5-minute slice, just some of the peptides that result from this LIS1 protein. We observe multiple glycide modified peptides in this context, and this is where it's really important to have the search parameters open with various miscleavage rates and enzyme and semi-enzymatic digest specificity. So this is a lysine 88, glycide modified peptide, very nice fragment ion spectra right here. We also see in that 1.5 minute time period, the wild type, many orders of magnitude more intense as we would expect, as well as lysine 76 right here. So all of these, as I said, are identified in real time. And what I want to drive at too is as well as getting information feedback in real time, PACER is more than just that. It's also the capability to drive the acquisition control. So PACER acquisition control, as we refer to that. And here's where that becomes really important. So when we look at the brain samples that we also ran using the cell signaling technology enrichment kits, we see greater proteome depth and much higher peptide coverage, and that's a function of the matrix that you're working with in itself. So on average, between 23,000 and 26,000 peptides that we identify. Now, I'll highlight here biological enrichment number 3, where for some reason it's a very low number of ubiquitination modified peptides that we see. So what's going on here? So we can ask that, and we can ask that in real time. We can say, is it the LC? Is it the mass spectrometer? Or is it actually the sample itself? So with this information in hand in real time, we automatically do a re-injection. We do that at a longer gradient. In this case, it's 45 minutes. We see in excess of 5,000 proteins, nearly 50,000 peptides identified. But here's the key. We still see a very small number of ubiquitin modified peptides, only 203. So that immediately tells us that the problem is at the level of the sample and not our LC or mass spectrometer. And that is the acquisition control parameters that you can use to decide to queue on further samples to be run. As I said, the timsTOF Pro data is 4-dimensional, and this adds complexity in how to view this data. So against traditionally using arbitrary units or intensities as your y-axis, we view the y-axis as the mobility or collisional cross-section term. What I'm showing you here is a phospholipid enrichment, and this is the distribution of all the ions that we see across the whole amount of time. We've been working with our software development team intensively, and we've built this PACER visualization functionality. And within there, you have all different kinds of features where you can slice your data accordingly. As it's 4th-dimensional, timsTOF Pro data has the advantage of providing you information at both the collisional cross-section level simultaneously with the MZ level, where traditional mass spectrometers will only provide you information at the MZ level. So one of the unique features that results there is something called mobility offset mass alignment. And that's a peptide that is isobaric or near isobaric in mass, but has a delta in its collisional cross-section in the gas phase. And those peptides can be searched for and highlighted effectively. Now, in the interest of time, I'm not going to go into the intricacies of how to define these MoMA groups, but what I will say is that these MoMA groups, and many thousands of them, as you can see, even at very tight tolerances, both at the MZ term, as well as the coelution term and the mobility term, are only visible with timsTOF data. So otherwise, all of these just look like chimeric spectra in the case of traditional mass spectrometers, where you don't have TIMS capability in the front end. As I said, you can focus in on specific things. In this case, we've decided to focus in on some very specific MoMA events, as being highlighted here by the red dots. You can toggle in, zoom in. I'm zooming in particular at this 655 ion. And what you'll see here is, even though it's isobaric in mass, near isobaric in mass, there's at least 2, if not 3 different mobility values or collisional cross sections. And you can see that by an extracted ion mobilogram, where there's 2 very baseline resolved unique feature sets. So those are definitely at least two unique peptides, which we will get a unique identification on each of them. One, because they have a unique mobility or collisional cross-section term, but also each will give a clean MS-MS spectra. And when I derive clean MS-MS spectra, that's a result of having trapped ion mobility separation in the front end of your mass spectrometer. So what TIMS does is it focuses the ions by size, space, and charge accordingly, and then it elutes them through the quadrupole into the TOF. And one of the results there is that the fragment ion spectra that come from timsTOF passive data is on average 4 times more pure. So what does that mean? What that means is, and this is work from Hannes Rust and his group, Joshua Charko, what they show is the number of co-fragmentation events that happen in timsTOF data is 4 times less than you would expect. And this is very much in line with what other people have showed. So this is actually work from my former advisor, Professor Roman Zubarov. And what they show is with the protonated lysine and arginine, even at a 1 Dalton mass window, about 90% of all your fragment ion spectra are chimeric in nature. And that is not the case when you do work with the timsTOF, because you have separation in the front end by collisional cross-section. This results also in some terrific selectivity and sensitivity applications that we can do with the mass spectrometer. So we've launched now DIA passive and PRM passive, and you can think of these two acquisition methodologies in the context of if you're defining the precursors that you're selecting by instrument control, or if the user is defining the precursors that they're selecting themselves in the case of PRM passive. And since we're visualizing the data by both the mobility or collisional cross-section and mass to charge, we can also move ions in that functionality. So in the context of DIA, as I'm sharing with you guys here, what you would really be used to in traditional DIA is opening up your quadrupole isolation window, getting all of this, what would be chemical noise, the 2+ ions that you're actually interested in, and then the 1+ ions, etc. And this creates a very noisy fragment ion spectrum. In the context of DIA passive, since you can move the ions as a function of both their mobility and mass to charge, you get that additional selectivity, and we can build this window across the 2+ ion cloud right here so that we're incredibly selective. And because it's passive enabled, parallel accumulation, serial fragmentation, that means that we're 4 times better at utilizing the ions as they come into the mass spectrometer. So all those things add up for us to be able to do extremely deep and quantitative proteomics. So what I'm showing you here is work done with the EvoSep1, where we use various gradients going as fast as 3 minutes. The 3-minute gradient results in the ability to run 300 samples per day. And even at 300 samples per day, we're still able to go 2,100 protein identifications deep into the proteome. And that results in nearly 10,500 peptide sequences. And of course, as you allow your gradient to go longer and longer, you'll observe more things. And the 60 sample per day methodology at 21 minutes is allowing us to go to about 5,200 proteins and nearly 40,000 peptide sequences in the DIA passive workflow. In the context of PRM passive, since the ions are trapped in the trapped ion mobility cell, and they have a unique dimension, which is the collisional cross-section or mobility and mass to charge, it allows you to multiplex at the PRM scale. So instead of just chasing one ion that you're interested in, you can actually chase 10 for every one that you would formally be able to do. So you can schedule those accordingly. And what I'm showing you in this upper panel here is how those are actually scheduled. And even you know, this looks like it's very messy, but since there are unique mobility terms and mass to charge, it allows you to multiplex. Additionally, since your ions are being focused in the context of space, size, and charge, you get this 30x boost in sensitivity. And we know that this group now is working with assays where they have about 600 targets in a 30-minute gradient that they're doing in PRM passive. I suggest please check out their publication. It's relatively recent, having come out in Analytical Chemistry. This is the work of Professor Lesueur's group at LIH in Luxembourg. And I really like this example here. So in the A panel, TIMS is off. And what happens there is you get these quasi-isobaric interferences, right? And this is unavoidable unless you have ion mobility in the front end. And the result is this nasty shoulder that you see here, which will definitely influence the quantitative capability once you go to do area under the curve integration of those particular peaks. But then you turn on the TIMS TIMS device, you separate ions by their ion or by their collisional cross-section term, enriching specifically for this guy, and that gives you this beautiful Gaussian distribution, which is going to give you much more accurate quantitation when you do that. So that's some of the unique capabilities. So now I want to see if I can revisit our demo lab right here, and here we are. So welcome PACER demo 50 sample per day was the method that we were running when we started this talk. And now I can go to the PACER box right here, and I will go away from the PACER key generator and into our projects folder. In that project folder, I made a folder where we can dump all of our data. In this case, it's this PACER view. And maybe I went to the wrong folder. Maybe it's in this one. And what I can share with you guys is the results, the real-time search results, where we see in this case 3,944 proteins identified in that 20-minute gradient, and about 26,768 proteins or peptides. I'm sorry. Right. So I will go back now to end my presentation and allow for some questions. So in summary, PACER provides run and done analysis, even under the most difficult search parameters, including variable modifications and post-translational modifications. You can visualize the data using the PACER visualization tool and pull out those mobility offset mass aligned features that are unique to timsTOF. TIMS and passive fragment ion spectra are substantially cleaner than other approaches. And then the selectivity and sensitivity of DIA passive and PRM passive provides high throughput and deep proteome analysis. And that will end my talk. I would be happy to take a couple of questions. Before that, I want to say this is teamwork. This is the contributions of many people. I'm thankful to all of them for their hard work. Okay. Let's see if there's any questions in there. What search algorithm? Okay, so the first question is, what search algorithm is currently used by PACER? So the search algorithm is a proprietary search algorithm We call it IP2 GPU. And it's compatible. Pacer is not currently compatible to other open source algorithms in its current discussion. Someone asked, Andy Quee asks, great talk. Could you comment on FAMES versus Bruker's ion mobility? I'm happy to do that. FAMES is effectively just a gas phase filtration. TIMS actually traps ions in the front end of the mass spectrometer and gives you a massive boost, 30x boost in the concentration because it focuses them by their collisional cross section. So in terms of FAMES and TIMS, there's no comparison. TIMS has much more advantages. And what I also should mention too is you'll never be able to derive a collisional cross section from a FAMES device where every single analyte, whether that be metabolites, small molecules, or peptides, derive a collisional cross section or mobility term using trapped ion mobility. I will allow for just a couple more in the interest of time. Actually, I would prefer to address the rest of these questions later in the interest of time. So with that, I will move on and I will stop sharing my screen to Dr. Shadi Ferdosi. Dr. Shadi Ferdosi is a scientist at Seer, and she is an experienced scientist at Seer, working in functionality between assay development, mass spectrometry, and proteomics data analysis. Dr. Ferdosi joined Seer very early after finishing her PhD at Arizona State University, exploring the diagnostic and prognostic capacity of blood, plasma, and serum glycans in different types of cancers. So we're very happy to have you here, Shadi. Thank you.
Hi everyone, my name is Shadi Ferdosi and I'm a scientist at Seer. Thanks to Bruker for their invitation to the launch seminar and giving us the opportunity to talk about our Proteograph platform, rapid and deep plasma proteomics at scale, coupled with the timsTOF Pro instrument. Plasma as a rich source of protein is commonly used in clinical profiling studies, but the large dynamic range of plasma proteome, which extends over 10 orders of magnitude, makes it challenging to interrogate deep into the plasma proteome. The current methods to address this challenge include depletion of top high abundant proteins and high pH peptide fractionation, which are laborious and low throughput, which limits the size of experiment and might lead to underpowered studies. To tackle this issue, Seer's nanoparticle technology comes in handy. When nanoparticles are introduced to blood plasma or other physiological matrices, they bind to proteins based on their physicochemical properties, such as shape, size, and charge, and form a layer on their surface called protein corona. So, by engineering nanoparticles with different physicochemical properties and using a panel of nanoparticles, we can capture proteins across the dynamic range of blood plasma based on their affinity to different nanoparticles and not solely based on their concentration. In our Proteograph assay, we process plasma sample by 5 nanoparticles in an automated way and analyze the resulted peptides by mass spectrometry. In this study, we've coupled the Proteograph with the Bruker timsTOF Pro. To test different gradients in DDA and DIA mode, we use the plasma pooled from healthy individuals, called PC3. Both the DDA and DIA data were processed through MaxQuant, filtering for 1% FDR at peptide and protein level. In DDA mode, the protein coverage and dynamic range coverage significantly improves by increasing the gradient length. Also, to give you a reference point, compared to neat plasma, we can identify and quantify about 5x more proteins using the Proteograph. And by using timsTOF Pro, we can identify about 30% more protein groups compared to the other standard mass spec platforms. In terms of precision, we can quantify peptides with median CV of less than 10% using any of the tested gradients. As we saw in the previous slide, in DDA mode we benefit from using a long gradient of 90 minutes, but that might not be efficient for a large plasma study, since for example a study with 500 clinical samples would take at least 5 months, and that's excluding the overhead and the potential instrument that downtime and all that, which could extend the timeline to 8 or 9 months. That's where DIA passive can be very beneficial. This method compared to DDA allows us to increase our protein identification and depth of coverage without increasing the length of gradient. As you can see, using a library-free approach to process the raw files on MaxQuant resulted in similar identification and dynamic range coverage compared to a timsTOF DDA-based spectral library, which is a huge advantage since that eliminates the need to generate a spectral library for each specific study. Also, in terms of precision, the library-free approach seems to be significantly better with median CV of less than 10% for 20 and 45-minute gradients. So for large plasma proteomics studies, using the 20-minute DIA passive method, we can significantly improve our throughput by at least 4 times with minimum loss of information. In conclusion, with the combination of Proteograph and timsTOF Pro, we can interrogate deep into the plasma proteome by using half of the typical peptide injection mass. Also, if this combination is used with the DIA passive method as well as the library-free approach on MaxQuant, that represents a unique opportunity for efficiency and enabling proteogenomics studies at scale. Thank you all for your attention. If you are interested in learning more about us, please connect with us and visit our posters or join our seminar at HUPO. Enjoy the rest of the conference.
Well, thank you, Shadi, for a fantastic presentation. We will open the floor to any that people might have on Seer's great proteograph technology. Let's see, how many? Yeah, would you please explain? First one is, would you please explain what are the 5 different corona nanoparticles? Are they different beads? I don't know what you can and cannot share, Shadi, but that's the question at you right now.
Thank you for the question. Well, the different particles have different chemistries. So based on their physicochemical properties, they can attract different proteins based on their affinity. So each of these particles will have different proteins on their corona. So when we inject the peptides resulted from these different particles, we can cover the dynamic range regardless of the protein concentration, but more based on their affinity to the different particles with the different chemistries.
Excellent. Thank you. Well, then we don't have any other questions, so we will now, in the interest of time, move to Professor Kirk Hansen. Thank you, Shadi. And I will say to everyone, I have a poster that I highlighted here that I'll be presenting in the afternoon. I would love to see anyone there that has questions. Dr. Ferdosi also has a poster with the Seer team that's being presented, and I'm sure that if you have any additional questions, they would love to address them at the poster session. So please don't be shy about reaching out. So now our highlight of the day is Professor Kirk Hansen. Professor Hansen is at the University of Colorado, Anschutz, where he's in the group of Biochemistry and Molecular Genetics. Dr. Kirk Hansen is a world-leading expert in the field of quantitative and extracellular matrix proteomics. Kirk received his PhD from Caltech and then would go on to do a postdoc at UCSF with Al Burlingame. Over the past decade, Dr. Hansen and his lab have invested in the development and translational application of analytical strategies for the detection and quantification of proteins in health and disease, with a particular focus on the extracellular matrix. And as the director of the University of Colorado School of Medicine Proteomics facility, Dr. Hansen has also collaborated with a range of scientists and clinicians on a diverse set of research and clinical projects. And this effort has resulted in more than 100 publications in less than 6 years' time. So yes, it's a pleasure to have you today, Dr. Hansen.
Thanks a lot, Chris. I'm gonna start by sharing my screen. Thanks everybody for attending today. Seems like there's a little bit of a lag here. Okay, and it looks like we're good to go. Okay, so today I'm going to talk about plasma proteomics, our efforts in plasma proteomics, and specifically focused around 2 COVID projects and a trauma project. This is a slide that I got from Angela De Alessandro, and this is just to show you that I think it's a little bit sort of underappreciated the years of life lost due to different disease states and trauma. Trauma is actually a pretty significant contributor to the total years of life lost in biomedical research, and yet, you know, one could argue, well, it's trauma, these are accidents, but there's a significant number of these cases where we have mortality that are thought to be preventable. I'm going to talk about that a little bit. I'm part of a research group that's focusing on trauma. And over the last several years, there's been a clinical trial called the COMBAT study. And the COMBAT study collected samples from trauma patients along a time series of 7 days. And in general, the injuries are categorized as blunt or penetrating injuries. And sort of actually, as I was looking for a COVID-omics figure or image of the team, I came across this image showing sort of the ranking of short-term mortality. And this actually comes from a life insurance company, so I take it with a grain of salt. But you have COVID, and then our 2 of our main causes of death associated with trauma. And in general, in this trauma project, we're interested in a number of things, but with respect to this data set and looking at the plasma, we're really interested in understanding with temporal resolution the body's response to trauma, and then to also identify novel meters of fibrinolysis, and I'll talk a little bit about that, and then also potential markers to guide resuscitation beyond what's used in the clinic right now. And so to kind of give you an outline, I'm going to start with a timeline. Of course, this was taken, this photo of my group and then Angela De Alessandro's group was taken before COVID. We're looking forward to getting back to social outings together again. And I'm going to talk about these plasma proteomic projects, and these are really carried out by primarily Monica in the lab. She has done all the work on the Bruker timsTOF instrument. I'm going to talk a little bit about one of Lauren's project characterizing essentially the insoluble structure of clots. And then a number of members from the team, Aaron, Brian, and Max, have helped with the data analysis efforts along the way. And so, you know, in the early days, we got kind of addicted to looking at the Johns Hopkins status site. And, you know, this is early March. In December, the instrument was installed. And now very early March, there were indications that in the US, we were headed for trouble. And then within you know, a matter of days, we had hot spots. One of those hot spots, of course, was New York. And that led to a collaboration that was spearheaded by Angelo looking at COVID samples, and that was submitted very rapidly after we ran those first samples. And during this whole process, we were gaining confidence in this analytical platform. And originally we started with a purely DDA workflow. And a lot of that had to do with, in our hands, we weren't getting significant better results with DIA passive, with DIA in general compared to DDA, at least at a level where we were confident in those results. And over time, we've transitioned. So I'm going to talk about that a little bit. So really what got us into the lab very quickly was Angelo found this paper, and in this paper, they had showed that you can inactivate SARS-CoV virus, CoV-1 in this case, and MERS, by doing essentially an organic extraction for metabolomics. And you could take that pellet and you could do proteomics on the pellet, and that's exactly what we did. And so we obtained approval to get back in the lab and start working May 7th. Monica was actually in the lab keeping the instruments up and running, and maybe or maybe not running some samples during that time. And ultimately, at very early on, we switched over to the EvoSep1. We chose to do the 30-minute, 30 runs per day method, 44-minute runs, about 41-minute gradients. And then we had found that the S-trap system from Protofy worked very well in our hands for plasma proteomics. And here I'm showing the 96-well plate, but actually at this stage, I believe we're using the single spin columns. And so here's what a typical run looks like. And I'm not going to talk much about this project. This was published, essentially this was submitted May 9th and then was up for publication in July. So really fast turnaround. And one of the things that I want to point out here is that we were seeing a lot of similarities with the trauma patient samples that we had run. We had run smaller cohorts of trauma patients in the past by plasma proteomics. We had done depletion. And really, I was never very comfortable running sample sets larger than maybe 24 or 40 samples. And so when we started to run 100 sample data sets, we went purely to an MRM-based assay using internal standards from QCONCATs. And so this was kind of the start of us getting back into plasma proteomics and a discovery platform. And even in these early days, our clinical collaborators were starting to think about pro-fibrinolytic agents. So specifically tissue plasmid activator as a clinical therapy, and they were starting the clinical trial at this time. And I mentioned there was a lot of similarities, and one of the things that really jumped out at us was that there was this crosstalk. There appeared to be crosstalk between complement activation and coagulation, and that's something that we had been exploring a little bit in the trauma patients. We noticed some of these signals, and so this definitely piqued our interest. The other thing that was starting to come out at this time is that the D-dimer levels in these patients was through the roof, and D-dimer is a breakdown product of fibrin, of course, and meaning that these patients were having thrombotic events. So a larger cohort, this time not extremely large, but over 100 samples between controls and COVID positive individuals was being initiated on campus. And here in Colorado, we have SomaLogic, and SomaLogic's in some ways a great platform for discovery proteomics and plasma. But I advocated that we used our mass spectrometry platform as well. And I'm really glad that we did. It led to some really interesting discoveries. Once again, we're using the same approach. And really some of the biggest changes between non-COVID and COVID infected individuals was the immunoglobulin response. And so really out of all the things that were significant and upregulated, I think all of them except for two were immunoglobulins, and then we had a couple markers of platelets. And so, for example, the platelet basic protein CXCL7 showed up as well. And when you started to do correlation with the seroconversion index, really that led to doing the seroconversion. That's the MSD assay that looks at, I think, 5 different COVID antigens. It's the one that's used for clinical 3 trials. We really saw very high correlation with these immunoglobulin chains and seroconversion. And that led to the idea that we would look at these correlations, and this was along a spectrum of when an individual, when a patient was converting from essentially a stage 1 response to a stage 2 response. And so this is really as you're switching from your activation of monocytes, dendritic cells, T cells, and natural killer cells to generating the beta cells that were creating the neutralizing antibodies. And of course, that's the part that you try to capture in a vaccination process without sort of this interferon signaling and the systemic complement activation, of course. And so that's all I'm going to talk about for that project, but you can actually go look at this data. I logged on this morning, it was a little bit slow, but you can actually look at all the different omics that were acquired from that data set, and there should be a preprint publication coming out soon from Joaquin Espinosa and his group. So back to the trauma project. And so after we had done these, what I would call, formerly medium-sized projects, now they're relatively small projects, we were really gaining a lot of confidence in our ability to do plasma proteomics and moving towards this combat data set where we had hundreds of samples. So let me quickly give you a background about the clinical problem. Of course, you've got your coagulation cascade, and the most key point here is that you eventually create thrombin. Thrombin cleaves the fibrinopeptides off of fibrinogen. Fibrinogen goes on to form polymers that are cross-linked by factor 13. Sort of the molecular process looks like this. You essentially get what's called the D region and the E region. The D regions align, come together, you make these fibers. Those go on to make larger fibers and branching, and then factor 13 cross-links and stabilizes the structure. But that's all in good at the site of an injury, but what happens is systemically you start having this same process occur, and so this can happen in the small capillaries of the lung, and that can create serious havoc. And so systemically you need a balance between the clotting process and breakdown of that clot, and that's driven primarily by plasmin. Plasmin identifies lysines on that clot. It sits down, something like thrombin activatable fibrinolysis inhibitor cleaves off those lysines, and then there's a whole range of events that block that process. And so this balance of this process is extremely important. And in trauma, when you have hypofibrinolysis, so shutdown of that fibrinolytic process, you essentially have very high mortality, and usually you get lung injury or ARDS and intravascular clotting that leads to death. Or you have hyper, and hyper is also equally bad, where you have overactivation of plasma or breakdown of that clot. And in particular, these are the patients that need massive transfusion. And there's been patients that have used up whole cities worth of blood supply in a single go. And then obviously it's very poor outcomes for patients as well. So how do we measure hemostasis or these clotting events in the clinic? Classically, they're measurements of prothrombin time, the Klaus assay for fibrinogen levels, and then D-dimers for the breakdown of clot. And so here, this figure, this would be an amplitude of clotting, and then along here would be the time axis. And then these are the classical assays. And the new assay that's being used, relatively new, would be maybe a little bit surprising to people how rudimentary this is that you actually put plasma sample in a cup. So this is an ex vivo clotting functional assay. This cup rotates, and then you have a torsion wire, and you can essentially measure the resistance of that's created on that wire. So mechanically, you're forming a clot. This time here tells you about how long it takes to form that clot, how strong that clot is, and then how quickly it breaks down. And usually you don't have complete breakdown of a clot, over a very long, multiple hours. And so there's these clinical signatures. And so the most important ones I'm going to show here are the fibrinolytic ones. And this is referred to by the fellows in the clinic as the stingray of death. And then you've got the death diamond. Neither one of these are good to see. And so as I was searching on the web, I actually came across this figure. This figure is for clinicians to make decisions, right? And so if you have a signature that looks like a wine glass, do nothing, give fresh frozen plasma. If you have a long activation time, if you have a weak plot, add platelets. give cryo, which is primarily factor 13 and fibrinogen, if you have this. And then if you have that stingray type figure, you give a lysine analog called TXA. And it turns out this assay drastically improves outcome, maybe not drastically, but it really significantly improves outcomes in the clinic. Overall bleeding, length of stay, mortality as examples. And so in terms of where we're going with this project is, on one hand, we're definitely interested in the hypothesis that this fibrin clot structure influences fibrinolysis, and that is difficult to study because it's an insoluble mass. And we've used our extracellular matrix methods to decellularize, essentially to remove all the protein except for that's covalently attached in the fibrin clot structure, and use chemical digestion with hydroxylamine to find, well, one, characterize the proteins present. And you can see here about 75% of the total protein is from fibrinogen itself, and then also identify the crosslinks. And so it was very gratifying to see, the only mapped crosslink that I was aware of before Lauren got her data sets was the crosslink responsible for the D to D region crosslink. This is the D dimer crosslink. And she, with the help of a visiting scholar, Zuzia Darula, identified that cross-linked form in either a mono or di-cross-linked species. And so here I'm showing that cross-linked species. Here are a couple other proposed sites of cross-linking where one side of the cross-link had been identified. And during the course of this action, we came up with many, many more candidates, and this has been published in JBC. And so then she's gone on to do sort of the kinetics of that cross-linking. But getting back to our plasma proteomics, there's also major influencers of the fibrinolytic process that are in the fluid phase in plasma. And sort of the classical ones are protease plasmin activated inhibitor 1 and 2, so SERPIN F1 and F2. TPA is your primary plasmin activator. There's also UPA and probably other proteases from neutrophils. And then your classic fibrinogen breakdown product is the D dimer. And so here's our setup again. This time we moved over to DIA. We're getting more confidence in our ability to run DIA. We have 144 patients, 828 samples across the time course, 101 healthy controls for a total of 985 runs, including the blanks. And this leads to a lot of data, as you can imagine. And we really struggled, honestly, with the data analysis. And originally we were using, a number of really powerful, really good software packages. We really like all of these packages, but we had serious limitations when it came to this large cohort. And so we were exploring the use of Spectronaut, and really it wasn't until the version 14 that came out at the end of last year that we were able to process this data. And then, there's a intermediate file type that's generated, and that can be generated as data is being collected. And so then you really have essentially creating the spectral library takes multiple days depending on your search parameters. But then we searched the whole data set in under a day. And when Aaron did this for the first time, I couldn't believe it because I was really anticipating a 3 to 4 week search time. Obviously, you create some very large reports, a CSV file of 8.7 gigabytes as the default. But luckily, the software has some really nice tools for modifying your outputs. And then also most recently, FragPipe and DIA neural network from Alexei and Vadim has now been adapted to work with DIA passive. I think they have a preprint coming out, and they were generous enough to share the software as of last week. And hopefully if I have a little bit of time, I'll show you some data off that. So when you first look at this trauma patient, here I'm showing healthy controls and then the time course across these patients. You can see these different phases. And what is kind of amazing to me is that we're throwing in patients that have injury scores from 1 to 75. And that, that's someone that fell off the back of their porch to someone that was in a, ran into the middle of the highway and was hit by 3 different vehicles. And so despite all of the differences in these patients, you see some very similar patterns. And that is, I've broken these down into the IL-1-like phase, acute phase response. Then there's a little bit slower IL-6 phase. And then there is some consumption. And one of the hypotheses has been that there's this consumptive coagulopathy. So essentially consuming all of your coagulation factors like the fibrinogens. The fibrinogens are never consumed past, I think, 30% of what they are in healthy controls. So it really gives us a lot of insight to some of the previous hypotheses that are out there. And then you also have a neutrophil phase, which is almost immediate. Platelet phase is the same. And then there's hemolysis. And so hemolysis is interesting because a lot of it does come from injury. You can see in the field before any blood products are given, that signal is already way up in a lot of patients. But now as you give blood products to patients, you also have hemolysis from stored blood products. So you need to use data reduction. How do you, to display this data and to try to wrap your head around it? And here I've done PLSDA. And so you get this fanning effect. And so this is the time course. You have healthy controls and you sort of have this offshoot in one dimension. Actually, it's in 2 dimensions, and then it fans back towards healthy controls within 7 days. And obviously we would have liked to have had a lot more sample than 7 days, because many of these patients aren't completely normalized at 7 days. But you can see here, if you now label these samples, a lot of them kind of hit in the same spot. And I just realized I'm going to be out of time. And so I'm going to kind of burn through some of this. You can actually use the metabolomics to see a lot of things like mannitol, and that tells us contributions of red cell hemolysis from the red blood cell units versus what we see in the patients. And so here you can see different patterns if you correlate to time. Okay, and this is actually the first pass of our data in Spectronaut, and I realized that we used overly stringent filters, because when I looked at the peptide level, we were filtering a lot of peptides from proteins that I know we cover very well. And so now, most recently, I've run semi-triptych, but also made the filters what I believe are more reasonable. We still need to validate that. And then I really opened them up to kind of see what the effect would be. This is the number of samples against number of proteins. And so here, this is saying that we essentially have 600 protein IDs in every single patient. And well, this is just patient samples, the 828, I believe. And so you can get candidates now by looking at correlations. And here's that pi1 again, that's being correlated against this lysis 30, the amount of lysis that occurs in this functional assay over 30 minutes. And so now what also comes up that wasn't as expected is tissue proteins, and specific muscle protein that is now correlating very strongly with the LY30. And so we can test those. And originally we had some candidates, they were, alpha globulin. We knew that hemolysis affects the coagulation phenotype quite a bit. And now you can get direct binding with Biochar. Now we're using MST. You can do functional assays. In this case, it attenuates plasmin activation. And then you can put it in a TEG cup with a healthy individual's plasma and see if you can induce the same type of response that you see in trauma patients. And so getting back to this clinical trial, these are COVID patients. You can see there's very little lysis, if any. And I'm going to just add that this, to me, looks like this cake holder. I don't know if there's a technical name for this, but I took a little bit of artistic liberty and added it to this figure that's on the internet. So here's the clinical trial. The big problem that we had, which was a very good problem to have, is that COVID went a little bit cold in May. And so enrollment was down a lot. And now enrollment is 13 controls and 17 TPA treatment patient groups. And overall, there's an improvement along essentially every metric, but we haven't achieved statistical significance yet. So that's ongoing. Lauren started to characterize the actual embolic clots that are found in some of these patients, and there's some interesting findings with the crosslinks. This took a lot of work. I didn't make any conclusions. Essentially, if you saw Dr. Steen's talk this morning, I want to echo essentially every conclusion that he made. I think he's had a very similar experience with the Bruker platform. It really has enabled us to do these plasma proteomic studies, and in the sake of time, I'm going to stop.
Thank you, Kirk. Thank you. We are pressed up against time. I will say that I'm sure, well, here's what I'll say. I will be at the poster session, and if there's any questions for Kirk that we don't get to today, I will definitely relay those questions to him specifically. You can find me at the poster session there. And then you can also find our Seer colleagues. They have a poster PO1.05 and PO8.07. So thank you all for attending today and have a wonderful rest of the conference.
Thanks, everybody.
Thanks, Kirk.